In a hospital, many patients were not recovering even after taking the same antibiotic. Doctor couldn't understand where was the real pathogen hiding. Then, microbiologists used the metagenomic technique. They analyzed all the microbial DNA together from the patient sample, and the result revealed a new antibiotic-resistant bacterium. After changing the treatment plan, and the patient slowly began to recover. These stories show that metagenomics in microbiology plays an important role in identifying invisible microbes, discovering new diseases, and improving treatment methods. Today, I will talk about metagenomics, an advanced field in microbiology. I am Suraiya Munia from the Department of Microbiology at Gono Bishwabidyalay. Now, let's know what metagenomics actually is. Metagenomics, also known as environmental and community genomics, is the genomic analysis of microbes by direct extraction and cloning of DNA from a microbial community. Microorganisms provide a culture-independent insight of microbial communities. It has emerged as a powerful centerpiece since most microorganisms can't grow in a pure culture, and that culturing cannot capture the full spectrum of microbial diversity. Now, let's know the history of metagenomics. 1970s. The first DNA sequencing methods were developed. Maxim Gilbert, chemical cleavage, and Sanger sequencing, chain termination in 1977. 1980 to 1990. Automation of Sanger sequencing led to large-scale projects like the Human Genome Project in 1990 to 2003. 2000s to present. Next generation sequencing technologies emerged, allowing rapid, high throughput, and cost-effective sequencing of entire genomes. At present, metagenomics continue to advancing along with other omic technologies. The field of metagenomics is rapidly advancing with improvements in sequencing technologies and bioinformatics tools. Now, let's know the classification of metagenomics. Number 1. Shotgun metagenomics. Number 2. Amplicon or targeted metagenomics. Number 3. Metatranscriptomics. Number 4. Metaproteomics. Number 5. Metabolomics. Two main types of metagenomics. Number 1. Whole metagenomics shotgun sequencing. Number 2. Targeted metagenomics. Whole genome sequencing in metagenomics is a technique that sequences all genetic materials like DNA present in an environmental sample, covering both culturable and unculturable microorganisms. It involves extracting total DNA from a sample sequencing. It uses high throughput technologies and then embedding and analyzing the sequences to identify organisms and their functional genes. Whole gene sequences helps to study microbial diversity, community structure, and functional potential, providing insight into metabolism, antibiotic resistance, and ecosystem functions. Targeted metagenomics has two parts. There are Number 1. Sequences-based approach, and Number 2. Functional-based analysis. Now, let's know more about them. Sequence-based approach. To retrieve specific genes for a pool of DNA by PCR or hybridization. For example, 16 ribosomal RNA, and 18 ribosomal RNA. Functional-based analysis. Functional-based analysis aims to discover genes with specific biological functions like enzyme activities, antibiotic resistance, biodegradation by searching metagenomic library for desired trait rather than identifying microbial species. Steps in functional-based metagenomic analysis. Number 1. Sample collection. Number 2. DNA extraction. Number 3. Construction of metagenomic library. Number 4. Functional searching. Number 5. Identification of positive clones. Number 6. Sequencing and bioinformatics analysis. Number 7. Functional characterization. Now let's find out the principle of metagenomics. To study all microorganisms in an environment by directly analyzing their DNA or RNA without the need to culture them in the laboratory. Some principles: sampling and DNA extraction, sequencing, bioinformatics analysis, genomic profiling, functional analysis, metagenomic analysis, all microbial DNA or RNA directly from environmental sample without culturing. Using high throughput sequencing and bioinformatics, it defines which microbes are present and what functions their gene perform. Now let's find out the importance of metagenomics. Helps identify culturable and unculturable microbes in any environment, reveal microbial diversity, community structure, and ecological rules, detects pathogens and track antibiotic resistance genes, supports research in human health, agriculture, environment, and biotechnologies, aids in discovering new enzymes, drugs, and bioactive compounds. Applications of metagenomics. Metagenomics helps identify novel microbial taxa, broadening our understanding of microbial diversity and ecosystems. Metagenomics identify functional genes and pathways, leading to enzymes discovering bioactive compounds and medical applications, such as environmental monitoring, human health, biotechnology, agriculture, bioremediation, industrial processes. It also helps discover new enzymes, antibiotic and bioactive compounds for biotechnology and medicine. Limitations of metagenomics. Metagenomics has some limitations. Metagenomics require complex data analysis, high cost, incomplete reference database, difficulty in embassy, less in DNA extraction or amplification, limited functional validation, contamination risk. It also can't always link gene to specific organisms or confirm actual biological function in the environment. Now let's learn about the future perspectives of metagenomics in microbiology. Metagenomics has transformed microbiology by embodying the study of microbial communities without the need for cultivation. It reveals microbial diversity, ecological rules, and functional potential, leading to discoveries in medicine, agriculture, environment, and biotechnology. There are some future perspectives, such as faster, cheaper sequencing, integration with other omics for deeper insights, better bioinformatics tools, expanded microbiome-based applications, support for environmental sustainability. We can be said in one word, metagenomics remains vital for exploring and utilizing the microbial world. Thank you so much for watching my presentation.